For decades, I mean, in medicine and certainly just in the popular imagination, we've relied on this really comforting mechanical metaphor for human appetite, haven't we?
Oh, absolutely. The old thermostat idea.
Right, exactly. Or, like a fuel gauge in a car. The idea is so incredibly simple. Your metabolic tank gets low, a little warning light dings on the dashboard of your brain.
You pull over, eat a meal, and the light switches off.
Yeah, and it implies a very clean, linear, binary system. You are either empty or full, starving or sated. But reading through the modern literature, that's just not true.
Not even close. It's an incredibly pervasive idea, though, stemming largely from the mid-20th century. Back in the 1940s and 50s, researchers like Hetherington and Ranson were doing these classic lesion studies in rats.
Right, the early brain mapping stuff.
Exactly. They would lesion the ventromedial hypothalamus, the rat would become massively hyperphagic and obese, and they concluded, well, we found the satiety center.
So they thought they found the off switch.
Spot on. And then they lesioned the lateral hypothalamus. The rat stopped eating and starved. And they declared, we found the hunger center.
The on switch.
Yeah, and we operated on this dual center hypothesis for a very long time. It was neat and tidy.
But looking at the foundational science we are exploring today, that dual center model seems not just outdated, but actually detrimental to how a clinician understands eating behaviors. The reality is profoundly messier.
It's incredibly messy.
Instead of a single fuel gauge, I've been trying to picture human appetite as, well, like a sprawling, highly chaotic international embassy. You have all these different diplomats arriving from completely different regions of the body.
I like that, the different physiological systems.
Yeah, you've got an envoy from the gut, an ambassador from your adipose tissue, a really strict delegate representing your biological clock. But then you also have these highly influential lobbyists from your emotional memory centers.
And don't forget the incredibly loud, disruptive delegates from your sensory organs. Your eyes, your nose, your tongue.
Oh, they're the loudest ones in the room. And every single one of those representatives is fiercely arguing over a single, highly contested policy decision, which is whether or not to initiate a meal.
And those negotiations really follow strict logic. Sometimes the gut envoy is screaming that the stomach is physically empty, but the emotional memory center steps in and issues an absolute veto because the organism is under intense acute stress.
Right, like when you get terrible news and completely lose your appetite.
Exactly. Or, the biological clock declares that it is 3 a.m, the kitchen must remain closed for metabolic repair.
But the visual cortex suddenly spots a leftover slice of chocolate cake in the fridge.
Precisely. And then the reward circuitry triggers an emergency override of the entire system, which perfectly grounds our discussion today in the core anchor of our field.
That psychiatry is fundamentally the study of how the brain, the mind, and experience interact.
And crucially, how that highly complex interactive process can go wrong. When we look at appetite through the lens of a distributed neural network rather than a single switch, we begin to understand why eating disorders and obesity are so biologically intractable.
They aren't just failures of a single gauge.
No, they are systemic dysregulations across a highly vulnerable, redundant network.
That brings us to our mission for this deep dive. Whether you are a psychiatry registrar prepping for your exams, a clinician looking to sharpen your reasoning around eating behaviors, or just a motivated medical student, this conversation is designed to be your companion.
Your companion to Chapter 1.17 of Kaplan and Sadok's comprehensive textbook of psychiatry.
Exactly. We are going to deconstruct the basic science of appetite. We'll start by defining the actual unit of measurement we care about, which is the meal.
Right, the meal is key.
And from there, we will explore the neurobiology of why we initiate eating, why flavor and hedonics keep us eating, and the precise mechanical and chemical signals that finally force us to stop.
And we really must connect all of that basic science directly back to clinical manifestation. We have to understand the healthy baseline, the homeostatic systems monitoring energy, and the hedonic systems assigning reward.
So that we can actually recognize the pathology when the system fails in conditions like anorexia nervosa and bulimia nervosa.
Spot on.
So let's start with that shift in the conceptual framework. If we are completely abandoning the idea of a central hunger switch in the hypothalamus, what exactly are we replacing it with? Because, for a busy clinician, the single switch model was at least conceptually clean.
It was easy to remember, yeah. But we are replacing it with a distributed network model that features what we call partially autonomous controls.
Partially autonomous. What does that mean in practice?
It means that control over eating is widely distributed across the brainstem, the hypothalamus, and the telencephalon, the higher cortical areas. Because the network is distributed, different nodes can act somewhat independently.
Meaning the different diplomats in our embassy have their own localized authority. So if I'm a psychiatrist prescribing a highly targeted pharmacotherapy, let's say a drug that blocks just one specific neuropeptide to suppress appetite, I shouldn't be surprised when the patient's weight loss suddenly plateaus after a few weeks.
That is the exact clinical takeaway. The system features massive evolutionary redundancy. If you pharmacologically mute one delegate in the embassy, the rest of the network simply adapts.
It just talks around them.
Pretty much. It upregulates other pathways and compensates for that silenced voice. You haven't changed the overarching policy, you've just created a temporary bottleneck that the brain quickly learns to route around.
Okay, so if the network is this complex and redundant, how do researchers even measure it? Reading through the studies, I was struck by how obsessively the field focuses on the meal.
Yes, the meal is everything in this literature.
But why look at a single meal? Why not just track a patient's total daily caloric intake or their weekly weight fluctuations? Isn't the macro data what matters for obesity or malnutrition?
Well, total caloric intake is a useful metabolic endpoint, sure, but it completely strips away all the behavioral phenomenology.
It misses the nuances.
Exactly. It tells you what happened over 24 hours, but it tells you absolutely nothing about how or why the brain made those decisions. The meal is both the functional and the biological unit of eating.
Functionally, what does a meal give us that a daily calorie count doesn't?
Concrete, real-time metrics of neural processing. When we look at the precise timing of a meal's initiation, the size of the meal, the microstructure of the eating rate.
Like, do they eat rapidly at first and then slow down, or maintain a steady, uncontrolled pace?
Right, and the interval between meals. All of that gives us a direct readout of how those competing embassy signals are being resolved minute by minute. An account of hunger entirely fails if it cannot explain the exact temporal moment a meal started and stopped.
And biologically, human beings, like most vertebrates, are naturally episodic eaters. We organize our intake into discrete bouts separated by fasting.
Precisely. And this is paramount for you as a psychiatrist. Eating disorders are fundamentally defined by disordered meals.
Right, not just disordered days.
Yes. Bulimia nervosa and binge eating disorder are diagnostically defined by the ingestion of abnormally large meals accompanied by a loss of control. Anorexia nervosa is defined by abnormally small meals or the absolute refusal to initiate a meal despite severe energy depletion.
The pathology manifests at the level of the meal. Okay, so the meal is our analytical battleground. Let's look at initiation. Why do we start eating?
Good question.
The textbook breaks the motivation of hunger down into 3 distinct constructs, and I think we need to carefully define these because they completely shift how we view a patient's drive to eat.
Yeah, hunger is not a monolith. We categorize it into 3 distinct motivational processes. The first is negative reinforcement hunger.
That sounds like the classic survival mode.
It is. This is the hardwired survival mechanism. You are in an aversive, deeply uncomfortable physiological state of nutrient deprivation, true cellular starvation, and your motivation to eat is driven entirely by the need to escape that negative, painful feeling.
So it is quite literally an escape behavior.
Yes. The second process is positive reinforcement related to hedonics. This is eating to obtain a pleasant sensory experience.
If you aren't starving, you just want something tasty.
Exactly. You are not trying to escape the pain of starvation. You are actively chasing the pleasure of a sweet, salty, or umami flavor on the tongue.
And the third?
The third is positive reinforcement related to post-ingestive consequences. This is eating to achieve that pleasant, sedated, almost anxiolytic physiological state that follows nutrient absorption and the release of gut peptides.
That feeling after a huge Sunday roast. But here's where I have to point out a massive disconnect between our evolutionary biology and our modern environment.
Oh, absolutely.
Because in affluent, industrialized societies, how often do we actually experience true negative reinforcement hunger?
Very rarely.
Right. If I think about my own daily routine, I fast for 10 to 12 hours overnight. When I wake up, my upper gastrointestinal tract is entirely empty. If negative reinforcement and the avoidance of starvation were running the show, I should wake up ravenous and consume a massive feast.
But you don't.
No. For me and for most people, breakfast is the smallest meal of the day.
And you have hit on a profound paradigm shift in how we understand modern feeding. For the vast majority of adults in food-secure environments, eating is almost entirely a lifetime of positive reinforcement learning.
We eat because it's pleasurable and habitual.
Exactly. We are driven by the anticipation of pleasure, the social context of food, and deeply ingrained conditioned associations with specific times of day. Innate homeostatic mechanisms crying out for survival are rarely the primary trigger.
Unless you're dieting or unwell.
Right. Negative reinforcement hunger is a state most people only encounter if they're engaging in severe, strict dieting, experiencing profound poverty, or suffering from a psychiatric condition like anorexia nervosa.
So if it isn't our biological fuel tank hitting absolute empty that triggers breakfast or lunch, what is pulling the trigger? How much of this is just the biological clock dictating the schedule?
Circadian rhythms exert a massive, often underestimated control over meal initiation. We have a master pacemaker in the brain called the suprachiasmatic nucleus, or SCN.
That's located just above the optic chiasm, right?
Yes, and the SCN has extensive, complex neural projections into the hypothalamus, specifically targeting areas that regulate feeding rhythms, like the subparaventricular zone and the dorsomedial hypothalamus.
But how much can the SCN really override our metabolic state, though? Like, if you are hungry, you are hungry.
Actually, it can override it completely. There are foundational forced desynchronization studies where subjects live in a laboratory for weeks under dim light, completely uncoupled from the sun.
Wow, that sounds intense.
It is. They put them on artificial 28-hour days. This allows researchers to separate the effects of internal circadian time from the effect of how long it has actually been since the person last ate.
So what does the data show?
It's unequivocal. Subjective hunger naturally dips to its lowest physiological point in the biological morning and peaks in the biological evening. This circadian drive operates completely independent of the duration of the preceding fast.
That perfectly explains the tiny breakfast phenomenon. The SCN is actively suppressing the drive to eat in the morning despite a 12-hour fast, likely because evolutionary pressures favored foraging and feeding later in the day.
Precisely.
But we can't ignore metabolism entirely. The sources do discuss blood glucose as a trigger, but it isn't the dramatic hypoglycemia that lay people talk about, is it?
No, it is much more subtle. The old glucostatic theory posited that cells literally ran out of glucose, triggering a starvation alarm.
Right.
But elegant studies in rats, like those pioneered by Campfield and Smith, showed that what actually precedes a spontaneous meal is a very tiny, highly transient drop in blood glucose. We're talking about a dip of maybe 10 to 12%.
That is barely anything. A 10% drop isn't going to compromise cellular function.
It doesn't compromise function at all. It appears the brain is not reading the absolute level of glucose. It is detecting the temporal dynamics of the decline.
The rate of change.
Yes, it is the downward slope that acts as an anticipatory signal. The brain is recognizing a pattern that usually precedes an energy deficit, and it initiates eating preemptively.
It is essentially algorithmic trading. The brain sees the stock market dip slightly, recognizes a historical pattern, and yells, "Buy now," before a hypothetical crash even occurs.
That's a brilliant analogy.
But glucose isn't acting alone. We need to introduce one of the most aggressive diplomats in our embassy, which is ghrelin.
Ghrelin. It is our primary endocrine instigator. It is a peptide hormone synthesized and secreted primarily by specialized endoendocrine cells in the mucosal lining of the stomach.
And its timing is very specific, isn't it?
Very. Its temporal pattern is unmistakable. Blood levels of ghrelin are high in the morning, they rise progressively and steeply during the intervals between meals, and they plummet rapidly the moment food enters the stomach and intestines.
So if you intravenously infuse ghrelin into a rat or a human, they will initiate a meal sooner, they will eat significantly more, and chronic infusion leads reliably to obesity.
Without fail.
But the nuance here is how ghrelin manipulates those different motivational hungers we defined earlier. It doesn't just make an animal want any calories, it specifically makes them want tasty calories.
Yes, the hedonic amplification is striking. Systemic administration of ghrelin in animal models stimulates the ingestion of highly palatable, sweet, or fat-rich foods much more potently than their standard laboratory chow.
So it makes the good stuff look even better.
Exactly. Furthermore, ghrelin facilitates conditioned place preference. If a rat receives a ghrelin injection in a specific distinctive chamber, it will subsequently prefer to spend time in that chamber, even when drug-free.
Oh, wow.
The brain associates the ghrelin surge with reward, indicating that ghrelin heavily ramps up positive reinforcement, hedonic eating.
But it isn't purely a hedonic drug, right? It also triggers the survival drive.
It acts on both. If researchers inject ghrelin directly into the third cerebral ventricle of a rat, bypassing the periphery and flooding the central nervous system, it dramatically increases how hard the rat is willing to work for a food reward.
In a progressive ratio test, right?
Yes. The rat might have to press a lever 5 times for a pellet, then 10 times, then 20. Ghrelin makes them willing to press that lever hundreds of times. They become highly, almost frantically motivated.
That's incredible.
This indicates it is also triggering that negative reinforcement, I must secure energy to survive, drive. Ghrelin acts on multiple brain loci, the hypothalamus for homeostatic drive, and the mesolimbic dopamine system for hedonic rewards simultaneously.
But the textbook introduces a relatively newly discovered molecule that complicates this picture entirely. LEAP-2, liver-enriched antimicrobial peptide 2. Why does a peptide related to the immune system matter for appetite?
LEAP-2 is a perfect illustration of why the single-switch model fails. LEAP-2 is an endogenous antagonist to ghrelin. It naturally binds to and blocks the ghrelin receptor, specifically the GHSR1A receptor.
And how do its levels fluctuate?
Crucially, its circulating levels in the blood run in the exact opposite reciprocal direction to ghrelin. When you eat a meal, your ghrelin drops, but your LATE2 levels rise.
Interesting.
And when a person gains adipose tissue and becomes obese, LATE2 changes to counter the effects of circulating ghrelin.
Wait, so the brain isn't just passively reading a thermometer that says, ghrelin is high, time to eat.
Exactly. The brain is constantly performing a complex stoichiometric calculation. It is high.
Right. Let's explore what happens next. Why do we keep eating once we start?
To understand the maintenance of a meal, we have to artificially separate the sensory pleasure of eating from the physiological fullness that follows. We have to isolate hedonics from homeostasis.
And in behavioral neuroscience, this is achieved through a classic, highly illustrative experimental paradigm called sham eating, right?
Exactly.
The gastric cannula experiments. Let's walk through exactly how this is done in the lab, because the mechanics paint a really vivid picture of how our brains are wired for reward.
Well, in animal models, researchers surgically implant a chronic gastric cannula, essentially a small sealable drainage tube placed directly into the stomach. When the cannula is capped and closed, the rat eats and digests completely normally.
But on test days,
On test days, the cannula is opened. When the animal drinks a liquid diet, the food enters the mouth, is tasted, is swallowed, travels down the esophagus, but then immediately drains out of the open cannula in the stomach.
so the food never actually accumulates in the stomach to cause mechanical stretch.
No, and it never empties into the duodenum to trigger chemical satiation signals. It creates a complete short circuit.
The brain gets all the sensory reward of the flavor via the cranial nerves, but absolutely zero post-ingestive feedback.
Precisely. And the results are dramatic. Without that gut feedback, rats will sham feed sweet solutions almost continuously for hours.
Just drinking nonstop.
The volume they consume is staggering, often far exceeding the physical capacity of a closed stomach. It beautifully isolates the sheer, unadulterated power of flavor hedonics. The sensory pleasure alone, without any caloric benefit, is enough to drive continuous massive consumption.
And we don't just see this in rats. The literature mentions human sham eating as well, which has direct psychiatric relevance.
Yes, in human paradigms, subjects are asked to chew highly palatable foods and then spit them out without swallowing. And clinically, this is a vital diagnostic window.
How so?
Studies have demonstrated that women diagnosed with bulimia nervosa display a significantly greater propensity and endurance for sham eating than healthy control subjects.
What does that tell us about their neurobiology, though?
It suggests a profound alteration in how their brains process hedonic reward. For a patient with bulimia, the flavor alone, the pure sensory input stripped of its caloric consequence, exerts a much stronger, more persistent behavioral pull.
So they just don't habituate to the pleasures quickly.
Right. Their reward circuitry is potentially hyper-reactive to the stimulus, or their top-down cognitive control is failing to habituate to the pleasure.
Which perfectly transitions to a phenomenon every single one of us has experienced, regardless of our psychiatric history. Eating in the absence of hunger, or EAH.
Oh, the dessert stomach.
Yes. You go to a massive holiday dinner, you eat a heavy roast, potatoes and vegetables, until you are physically uncomfortable. You swear you couldn't possibly ingest another calorie. And then the host brings out a rich chocolate pudding.
And magically, you have room.
Exactly. You find the physical room to eat it. How is the brain overriding that profound physical fullness?
Well, the dessert stomach is the colloquial term for a deeply ingrained neurological mechanism called sensory-specific satiety. It is heavily driven by the evolutionary need for dietary variety.
So as you eat that large roast dinner,
The reward response in your brain to those specific savory, umami, and salty flavors begins to habituate. The neurons literally fire less robustly. Your brain signals satiety, but it is a satiety specific only to that flavor profile.
the brain is bored of the beef.
That's a good way to put it. But when the novel, highly palatable sweet flavor of the chocolate pudding is presented, it bypasses that habituated circuitry. The sweet flavor activates fresh, unhabituated reward pathways.
The variety itself is driving the eating.
Yes, variety itself is a powerful hedonic control of eating. This is an evolutionary adaptation. Foraging for a wide variety of flavors ensured our ancestors obtained a diverse profile of micronutrients and amino acids.
But in the modern world, it makes us incredibly vulnerable. It's why you eat vastly more at an all-you-can-eat buffet with 20 different dishes than you would if you were served a massive trough of just your favorite meal.
The constant novelty hacks the sensory-specific satiety mechanism.
But let me ask you about these flavors. Are we born loving chocolate pudding and disliking broccoli, or is this all learned?
Aside from two major exceptions, almost all human food preferences are learned. We are born with an innate, hardwired preference for sweet tastes because sweetness is a highly reliable evolutionary marker for safe, dense carbohydrate energy.
And the other exception.
We are also born with a hardwired aversion to intensely bitter and sour tastes because those are reliable botanical markers for alkaloids, poisons, or bacterial spoilage. Beyond those innate reflexes, the rest is a product of conditioning.
How does that conditioning actually work? If a flavor is neutral, how does it become a craving?
The primary mechanism is flavor-nutrient conditioning. This is a form of Pavlovian learning. Your brain continuously pairs the specific, conscious sensory experience of a flavor, the conditioned stimulus, with the deeply rewarding, unconscious, post-ingestive metabolic effects that follow nutrient absorption.
The unconditioned stimulus.
Exactly. If you eat a novel food with a strange flavor, and 20 minutes later your brain receives a surge of glucose and a calming release of gut peptides, your brain retroactively tags that flavor as good.
This is how we overcome our innate fear of new foods or neophobia.
Precisely. It is how complex cultural cuisines develop. You learn to love the burning sensation of chili peppers, the pungent aroma of fermented cheeses, or the bitter bite of black coffee.
Not because those flavors are innately pleasing.
No, but because your central nervous system has meticulously tracked the positive physiological, pharmacological, and social rewards associated with them over years of exposure.
But I imagine the baseline hardware we use to perceive those flavors matters too. We don't all experience the physical sensation of taste with the same intensity.
We don't. There is vast anatomical and genetic variation in taste perception. Consider the fungiform papillae, the small mushroom-shaped structures on the anterior surface of your tongue that house your taste buds.
Right.
The density of these papillae varies wildly between individuals. People with a very high density are often termed supertasters.
And is this just random variation?
No, there are distinct demographic patterns. Interestingly, fungiform papillae density and the consequent intensity of taste perception is generally higher in females than in males.
Oh, really?
Yes. Furthermore, genetic variations, such as the alleles for the TAS2R38 bitter taste receptor gene, dictate how intensely an individual perceives specific compounds.
So an individual with high papillae density might literally experience a piece of milk chocolate as a much more intense, overwhelming sweet reward than someone with lower density.
Yes, and the clinical implication is significant. This individual anatomical difference in taste capacity directly correlates with the intensity of hedonic pleasure derived from food.
That makes sense.
And robust studies show that this heightened hedonic responsivity correlates with a higher body mass index, or BMI, over a lifespan. Your literal tongue anatomy can tonically increase your meal size by amplifying the positive reinforcement signals to the brain.
That is a staggering integration of macroanatomy and behavior. All right, we've initiated the meal. We are riding the wave of flavor hedonics, learning, and sensory-specific satiety. Now we must address the most critical regulatory step.
Satiation.
Yes, why do we eventually stop eating? Satiation. What is the physical emergency brake that finally overrides the dopamine hit of a delicious meal?
Satiation is not a passive process of simply running out of food or getting tired of chewing. It is an active, aggressive neurological intervention. The brain must receive specific, potent stop signals from the periphery to terminate the meal.
And the first line of defense in that process is purely mechanical, isn't it? Gastric mechanoreception.
Correct. We are talking about the physical stretching of the stomach wall. As ingested food and liquid fill the stomach, the physical distension activates specialized mechanoreceptors.
These are the IGLEs.
Yes, primarily intraganglionic laminar endings, or IGLEs, located in the muscular layers of the stomach wall. As the stomach stretches, these receptors fire electrical action potentials up the afferent fibers of the vagus nerve, traveling directly into the brainstem.
I read an interesting clinical application of this. It's why dietitians tell you to eat soup rather than drink water with a dry meal.
Exactly. If you drink a glass of water alongside dry food, the liquid empties from the stomach into the duodenum very quickly, reducing the gastric volume.
So the stretch goes away fast.
But if that same amount of water is incorporated into the food matrix, like in a thick soup or a stew, the gastric emptying rate is significantly slowed. The physical stretching of the stomach is maintained for a longer duration, which sustains the vagal mechanoreceptive firing and prolongs the feeling of fullness.
But the physical stretch is just the brute force mechanism. The intestines are also doing complex chemical analysis of the food as it arrives, right?
Absolutely. The gastrointestinal tract is arguably our largest sensory organ. As chyme, the partially digested food enters the small intestine, specialized enteroendocrine cells sample the nutrient content.
And they release a cascade of chemical messengers.
We see the release of glucagon-like peptide 1, peptide YY, and the absolute undisputed star player of satiation cholecystitinin, or CCK.
I want to spend time on CCK, because the textbook holds it up as the absolute gold standard for a satiation signal. Why is CCK so highly regarded compared to the dozens of other gut peptides we've discovered?
Well, CCK is uniquely established because it is one of the very few peptides that fulfills all the rigorous scientific criteria required to prove it acts as an endogenous physiological signal, rather than just a pharmacological artifact.
So we know it's naturally doing the job.
Yes. CCK is secreted rapidly by the eye cells in the mucosal lining of the upper small intestine in direct response to the presence of fats and proteins.
So if I eat an avocado or a piece of chicken, CCK floods my system. How do we know it actually causes satiation in humans and doesn't just, make us feel nauseous so we stop eating?
That is the crucial distinction. If you intravenously infuse human volunteers with CCK at doses that perfectly mimic the normal physiological spikes seen after a meal, they will reliably consume a smaller meal.
Okay.
And crucially, they do not report feelings of nausea or visceral illness on standardized scales. They just report feeling pleasantly full sooner.
But the real definitive proof usually comes from blocking the signal.
Yeah.
Right? The antagonist studies.
Exactly. And this is where CCK separates itself from the pack. If you administer a selective CCK receptor antagonist to a human being, the drug binds to the CCK receptors on the vagus nerve and blocks the brain from hearing the CCK being produced by the gut, the individual's meal size significantly and reliably increases.
They just overeat.
Yes. We do not see this clean, robust, bidirectional effect for almost any other gut hormone in human trials. Furthermore, animal models with genetic knockouts of the CCK receptor, like the OLITF rat we will discuss later, exhibit hyperphagia and massive obesity.
CCK is the primary biochemical brake pedal.
It really is.
Okay, so we have the physical stretch of the stomach and we have the chemical release of CCK from the intestines. Both of these signals need to get to the brain, and you mentioned they travel up the vagus nerve.
They do.
Before we go further, we need to define the destination. Where exactly in the brain are these vagal fibers going?
The vagus nerve fibers project up into the hindbrain and terminate in a specific cluster of neurons called the nucleus tractus solitarius, or the NTS. You can think of the NTS as the brainstem's grand arrival hall for all visceral sensory information.
So the NTS receives the stretch signal, and it receives the CCK signal. Do they just add up? Or is there something more complex happening in this arrival hall?
This is where the neurobiology becomes truly elegant. The integration is not merely additive, it is highly synergistic.
Synergistic how?
Well, if researchers isolate an individual vagus nerve fiber in an animal model and measure its electrical firing rate, they observe something remarkable. If you physically stretch the stomach with a balloon, the fiber fires at a low, moderate rate.
Right.
If you infuse a small amount of CCK without stretch, it fires at a similarly low rate.
But if you do both,
Yeah.
if you stretch the stomach and infuse the CCK simultaneously, mimicking a real meal.
The vagal firing rate absolutely explodes. The resulting action potentials are vastly greater than the sum of the two individual stimuli. 1 + 1 doesn't equal 2 in the NTS. 1 + 1 = 10.
That's a massive amplification.
Yes, the system is designed to require concurrent validation. The brain wants confirmation from multiple modalities, mechanical volume and chemical nutrient density, before it fully commits to shutting down the powerful biological drive to eat.
And the vagus nerve itself, this massive neural highway connecting the gut to the brain, is hiding some fascinating structural secrets. The sources highlight recent cutting-edge optogenetic work in mice regarding vagal laterality. Can you explain what laterality means in this context?
We traditionally viewed the sensory vagus nerve as a uniform conduit. The vagal sensory cell bodies reside in two bundles located just outside the brainstem, known as the left and right nodose ganglia.
Right, the left and right bundles.
For a long time, we assumed they did the exact same job. But recent, highly sophisticated research utilizing optogenetics, where scientists can use light to selectively turn specific neurons on and off in living animals, has revealed a functional asymmetry.
They have completely different jobs.
Yes. It appears that activating the afferent fibers originating specifically from the left nodose ganglion strongly mediates satiation. It delivers the firm, stop-eating feedback.
And the right.
However, activating the fibers originating from the right nodose ganglion seems to mediate reward, appetition, and the drive to seek out food.
That is mind-bending. The left lane of the highway is carrying the brakes, and the right lane is carrying the accelerator.
It forces us to reconceptualize the gut-brain axis entirely. The vagus is not just a single wire delivering a fullness score. It is a multi-lane, bidirectional highway carrying highly nuanced, anatomically segregated, and sometimes opposing instructions to the brainstem.
All right, we have covered the initiation, maintenance, and satiation of a single meal. But eating isn't just about one meal. The body must regulate energy over months and years.
The macro level.
Exactly. Let's zoom out to the macro level. We need to talk about long-term energy homeostasis and the hormonal battleground of the hypothalamus. And that means we have to address leptin.
Yes, we must.
Let me ask the question that every frustrated clinician and dieting patient has asked since the 1990s. If leptin is a hormone secreted by our fat cells, specifically to tell the brain we have plenty of stored energy, why doesn't it work?
It's a great question.
Why doesn't a person with excess adipose tissue, and therefore massively high leptin levels, just naturally stop eating? Why didn't leptin injections cure the obesity epidemic?
It is the great paradox of modern endocrinology, and to understand it, we must look at the system through the lens of evolutionary biology. The fundamental truth about leptin is that its signaling is highly asymmetrical.
Asymmetrical in what way?
It evolved almost exclusively to defend the organism against starvation, not to protect against obesity.
Explain that asymmetry. How does the brain interpret a drop in leptin versus a spike in leptin?
Consider the selective pressures on early hominids. Starvation was a constant, imminent, and lethal threat to survival and reproduction. Obesity, as we know it today, was virtually non-existent.
So we didn't need a defense mechanism against it.
Precisely. Therefore, the neural circuitry evolved to be exquisitely, fiercely sensitive to a deficit in energy stores. When an individual loses fat mass, circulating leptin levels drop below their biological set point.
And the brain panics.
The hypothalamus detects this drop and interprets it as an absolute metabolic emergency. It triggers overwhelming, unrelenting negative reinforcement hunger and simultaneously depresses thyroid function and cellular metabolism to ruthlessly conserve energy.
The alarm bells are deafening when leptin drops. But what happens when the individual finds a surplus of food, gains excess fat, and leptin levels soar above the baseline?
The brain largely ignores the surplus signal. Once leptin levels are sufficient to reassure the hypothalamus that starvation is not currently imminent, adding more leptin, whether synthesized by excess visceral fat or injected via a syringe, has very little physiological effect on suppressing appetite or increasing energy expenditure.
That's fascinating.
The brain lacks a strong evolutionary imperative to defend against a caloric surplus. Its evolutionary logic is basically, excellent, we have secured excess energy to survive the famine that will inevitably arrive tomorrow.
So we are biologically blind to surplus. That is incredibly frustrating, but deeply logical. So leptin monotherapy is essentially useless for a generally obese patient because their brain is already ignoring their high endogenous leptin.
Exactly. It only shows clinical promise in two scenarios. First, for patients with extremely rare genetic mutations where they physically cannot produce leptin, they are functionally starving despite massive obesity.
And the second scenario?
potentially as a maintenance therapy for individuals who have already dieted down, lost significant fat mass, and are trying to prevent the starvation rebound response caused by their suddenly depleted leptin levels.
So where exactly in the brain is this leptin signal being received and processed? Let's take a tour of the arcuate nucleus, or the ARC, located at the base of the hypothalamus. Reading the neurobiology here, this seems to be the absolute epicenter of the network's conflict.
The ARC is the primary hypothalamic sensor for circulating metabolic hormones because it sits very close to the median eminence, an area where the blood-brain barrier is somewhat permeable.
And what's inside the ARC?
Within the ARC, we have two highly specialized, distinct, and directly opposing populations of neurons sitting right next to each other.
Let's call them Team Stop and Team Go. Who makes up Team Stop?
The stop-eating team are neurons that co-express a precursor protein called POMC and CART. Crucially, the POMC is cleaved to produce alpha-melanocyte-stimulating hormone, or alpha-MSH.
And what triggers them?
These POMC neurons are strongly stimulated by leptin and insulin. When energy stores are high, these neurons fire, releasing alpha-MSH to suppress appetite and increase energy expenditure.
And sitting right beside them is Team Go.
Yes. Team Go consists of neurons that co-express AGRP, neuropeptide Y, and the inhibitory neurotransmitter GABA. These neurons act in direct opposition.
So they are inhibited by leptin.
They are potently inhibited by leptin and insulin, but powerfully stimulated by ghrelin from the gut. When energy stores are low, leptin drops, removing the inhibition, and these neurons fire rapidly to drive intense feeding behavior.
So the ARC is reading the hormonal environment and deciding which team gets to play. But how do these teams actually enact their policies? Where do they send their signals?
Both the POMC neurons and the AGRP neurons send their axonal projections outward to second-order target areas, primarily the paraventricular nucleus and the lateral hypothalamic area.
And this is where the conflict happens.
It is. And here is the specific molecular mechanism that every clinician must master. Both alpha-MSH from Team Stop and AGRP from Team Go project to and compete for the exact same receptor on those downstream target neurons.
They're literally fighting over the same steering wheel.
That is an excellent mechanical analogy. The steering wheel in this case is the melanocortin-4 receptor, or the MC4R. It is a G protein-coupled receptor.
How does alpha-MSH drive it?
Alpha-MSH acts as an endogenous agonist. It binds to the MC4R, turns the wheel, activates the receptor, and forcefully suppresses appetite.
And AGRP?
AGRP, however, is a fascinating molecule. It acts as an inverse agonist or a functional antagonist. It binds to the exact same MC4R, blocking alpha-MSH from attaching, and actually drives the receptor's baseline activity below zero,
Which violently ramps up appetite. This sounds like a critical choke point. If this is the primary steering wheel for the homeostatic system, a single point of failure here would be catastrophic for the patient.
And we see exactly that in clinical genetics. Loss of function mutations anywhere in this MC4R pathway, whether in the POMC gene, the receptor gene itself, or the processing enzymes, are the most common recognized cause of monogenic human obesity.
Because the brake is completely gone.
If that MC4R receptor is structurally broken or unable to receive the alpha-MSH signal, the brain's primary homeostatic brake pedal is permanently disabled. The patient experiences unrelenting, severe hyperphagia from infancy.
And understanding this basic circuitry isn't just academic trivia. It leads directly to profound, targeted interventions.
Absolutely. The translation from bench to bedside here is remarkable. We now have targeted pharmacotherapies, such as setmelanotide. This drug is a highly specific MC4R agonist.
So it replaces the missing signal.
Yes. It was recently approved specifically for patients with genetically confirmed mutations upstream in this pathway. We're literally designing synthetic molecules to act as artificial alpha-MSH, bypassing the broken POMC neurons to manually flip that specific receptor switch and restore satiety.
That is the ultimate validation of learning the basic science. Okay, we've covered the brainstem's handling of the gut signals and the hypothalamus's handling of the long-term metabolic hormones. But as we established earlier, human eating is largely driven by hedonics and pleasure.
The reward system.
Exactly. Let's move up the neuroaxis to the telencephalon, the higher brain regions. We need to map the brain's hedonic hotspots.
If the hypothalamus is the strict accountant meticulously calculating caloric deficits and leptin levels, the telencephalon, specifically the mesolimbic reward circuitry, is the aggressive marketing department.
Constantly trying to convince the organism that food is irresistibly appealing and worth pursuing regardless of the budget.
Spot on.
Let's start with the subcortical nodes. When I take a bite of that rich chocolate pudding, What is happening in the nucleus accumbens?
The nucleus accumbens, particularly the medial shell region, is flooded with dopamine. This dopamine release is highly precise and dose-dependent.
How so?
In microdialysis studies in rats, if you present them with different concentrations of a sucrose solution, the volume of dopamine released in the nucleus accumbens scales linearly with the concentration of the sugar. A 20% sucrose solution releases massively more dopamine than a 5% solution.
So the accumbens is encoding the exact reward value of the stimulus. But we need to clarify something crucial about dopamine because it's often misunderstood in pop psychology as the pleasure chemical.
Yes, that is a critical distinction developed extensively by researchers like Kent Berridge. Dopamine in the nucleus accumbens does not mediate the subjective sensation of liking the food, the actual conscious pleasure.
So it's not the pleasure itself. What is it?
Instead, dopamine mediates wanting. It assigns incentive salience to the food cue, generating the intense motivation, the craving, and the drive to seek it out.
If dopamine is just the craving, where does the actual pleasure come from? What generates the liking?
For that, we look to adjacent structures, particularly the ventral pallidum, or VP. The VP receives massive projections from the nucleus accumbens, and its hedonic functions rely heavily on endogenous opioid and endocannabinoid signaling rather than dopamine.
And there is a truly fascinating lesion study about the ventral pallidum that proves its role in generating pleasure. Can you detail the effective reactivity experiments?
This is one of the most striking findings in effective neuroscience. Researchers can measure liking in animals by observing their innate facial expressions. Sweet tastes elicit relaxed lip smacking, while bitter tastes elicit aversive gapes and head shakes.
Right.
If researchers physically lesion or damage the caudal portion of the ventral pallidum in a rat, the results are profound.
Does the rat just become indifferent to sugar? Does it lose the ability to care?
It is far more extreme than indifference. If you place a drop of highly concentrated sugar water directly into the mouth of a rat with a ventral pallidum lesion, it does not display the normal lip smacking.
What does it do?
Instead, it displays active, visceral, aversive facial reactions. It gapes and flails as if you have just fed it battery acid or a highly toxic bitter alkaloid.
Wait, the physical sugar hasn't changed. The taste receptors on the tongue are still sending the sweet signal.
Exactly. But the ventral pallidum is missing. This implies that the VP is not just a passive relay station passing along a sweet message.
It's actually creating the experience.
Yes, the ventral pallidum actively, chemically generates the subjective, conscious experience of pleasure. It transforms the sensory data into an affective state. Without the opioid signaling in the VP, the brain's default, unmodulated reaction to a potent novel stimulus might actually be aversive. The VP constructs the feeling of, this tastes good.
That is incredible. The brain manufactures the pleasure. Let's move higher up to the cortical nodes, the highly evolved outer layers of the brain. Where does the orbitofrontal cortex fit into this marketing department?
The orbitofrontal cortex, or OFC, acts as a highly sophisticated real-time value monitor. It integrates the sensory pleasure of the food with your current physiological state and cognitive context. Functional magnetic resonance imaging studies in humans show this beautifully.
How does it react to the dessert stomach phenomena?
It is the neural representation of it. If a human subject is fasted and hungry and you place them in an fMRI scanner and let them smell or taste a highly palatable food like chocolate, specific regions of their OFC will fire intensely. The value of that stimulus is extremely high.
But then you feed them.
Right. You take them out, let them eat that exact chocolate until they report being completely satiated and physically full. You put them back in the scanner and present the exact same chocolate smell or taste again.
And what happens?
The sensory input is identical, but the fMRI shows that the OSC firing in response to that specific food is drastically reduced, almost silenced.
The food hasn't changed, but its objective value to the organism in that precise physiological moment has plummeted.
Exactly. But, and this is the key to sensory-specific satiety, if you then present the smell of a completely novel food, like a savory pizza, the OFC will light up again. It constantly updates the subjective reward value of specific stimuli based on immediate biological feedback.
All right, we have meticulously built this incredible network. We have the gut, the vagus, the NTS, the hypothalamic ARC, and the telencephalic reward hotspots. But the network doesn't exist in a vacuum. Let's examine what happens when we introduce outside variables. We're moving into Section 6, modulators of appetite, things like exercise, learning, and sex hormones.
This is where it gets very clinically relevant.
Let's start with exercise, because the clinical reasoning here goes far beyond just burning calories. I want to delve into the animal models, specifically the OLTF rats. What makes these rats so special?
The OLTF rat, which stands for Otsuka Long-Evans Tokushima fatty, is a profoundly valuable genetic model in obesity research. Due to a natural genetic mutation, these rats are born completely lacking functional CCK1 receptors.
So they are entirely missing the brain's ability to hear the primary satiety signal from the gut? They have no chemical breaks.
Correct. Consequently, from a very young age, their meals are abnormally large. They exhibit chronic hyperphagia, they vastly overeat, and they reliably develop severe obesity and type 2 diabetes. Their baseline network is fundamentally broken.
So how did researchers use them?
Researchers utilize this model to see what happens when you introduce exercise as a network modulator.
What happens if you take an adult, obese OLTF rat and put a running wheel in its cage?
The adult rat will run. And remarkably, its hyperphagia will decrease, its food intake will normalize, and it will lose significant weight. The physical activity somehow overrides the broken CCK satiety signaling.
That seems like a massive win for the just exercise more prescription.
It is, until you remove the wheel. The very day you lock the running wheel, preventing the adult rat from exercising, its hyperphagia immediately returns in full force, and it rapidly regains all the adiposity.
Oh, wow. So it's not a cure.
The exercise was merely a temporary active patch. It suppressed the symptoms, but it didn't fix the underlying neural circuitry.
But here is where the developmental neurobiology gets genuinely exciting. What if you intervene earlier? What if you give the wheel to a juvenile rat?
This is the critical paradigm-shifting finding. If researchers provide the OLTF rat with access to a running wheel before puberty, during the critical developmental window of adolescence, while its neural circuits are still plastic and forming, and then they completely remove the wheel when the rat reaches adulthood.
The protective effect is permanent.
Yes.
Wait, even without the wheel in adulthood, they don't get fat?
Exactly. Their food intake remains normalized, their meal sizes remain small, and their body weight stays low for the rest of their lives, completely independent of ongoing exercise.
Hold on, you can't just say the exercise fixed them. What actually physically changed in the rat's brain during that prepubertal window to permanently correct a genetic lack of CCK receptors?
It is a profound example of experience-dependent neuroplasticity. While the exact molecular cascade is still being mapped, the evidence suggests that the stress and metabolic demands of the early exercise physically altered the architecture of the developing brain.
Remodeling the network itself.
Yes. It likely involves the upregulation of neurotrophic factors like BDNF in the hindbrain. This early intervention forces the vagal afferent terminals in the NTS to remodel and form new compensatory synaptic connections.
Oh, incredible.
It might also induce epigenetic changes that permanently alter the expression of other satiety peptides. The exercise didn't just burn calories, it acted as a structural engineer, literally hardwiring a compensatory satiety pathway into the brainstem while the concrete was still wet.
That has staggering implications for how we view physical education and childhood activity. It isn't just about managing a child's current weight. It is about permanently organizing their hypothalamic and brainstem networks for a lifetime of metabolic resilience.
Absolutely.
Let's pivot to another massive, highly relevant modulator, the reproductive axis. As clinicians, we have to recognize demographic realities and their profound sex differences in appetite regulation.
The interaction between sex steroids and appetite networks is very significant, yet historically it has been frustratingly overlooked in basic science, which defaulted for decades to using only male animal models.
How does it manifest in women?
In women and female mammals generally, spontaneous food intake fluctuates reliably and predictably across the ovarian cycle.
What does that cycle look like in terms of caloric intake?
During the follicular phase, and particularly the periovulatory phase, when circulating estrogen levels reach their peak, women spontaneously eat less. Their meal sizes decrease.
Are they doing this consciously?
This is rarely a conscious dietary restriction. It is a subconscious, hormonally driven reduction of perhaps a few hundred kilocalories per day.
Why does estrogen do that? Is it acting on the hypothalamus or the brainstem?
Both. But its action in the brainstem is deeply fascinating. Estrogen receptors are heavily expressed on the neurons in the NTS, that grand arrival hall we discussed. Experimental evidence suggests that estrogen actually sensitizes the NTS to vagal inputs.
So it turns up the volume on the gut stop signal.
Precisely. So when estrogen is high, A normal pulse of CCK feels substantially more satiating. Smaller meals feel disproportionately filling.
But what happens later in the cycle?
Following ovulation, during the luteal phase, estrogen levels drop and progesterone levels rise. Progesterone essentially counteracts the inhibitory effects of estrogen, often by downregulating the estrogen receptors.
And the volume gets turned back down.
With that sensitization removed, the satiety threshold rises, driving food intake back up. This frequently presents clinically as premenstrual cravings or increased meal sizes.
And beyond the fluctuating hormonal cycle, there are baseline, hardwired sex differences in hedonic preference, correct? We touched on the fungiform papillae earlier.
Yes, there is a clear biologically driven sex difference in sweet avidity. Across numerous mammalian species, including humans, females exhibit a stronger behavioral preference for highly concentrated sweet flavors than males do.
Why would that be evolutionary advantageous?
This is likely an evolutionary adaptation to ensure adequate adiposity for the massive metabolic demands of gestation and lactation. However, in a modern environment overflowing with engineered ultra-processed sweets, this ancient female typical biological drive can become an independent, tonically active risk factor for overconsumption.
All right, this brings us to the ultimate synthesis, the most clinically critical part of our deep dive. Section 7, Psychiatric Eating Disorders. We have mapped the healthy network. We know how it initiates, maintains, and terminates a meal.
We know how it is modulated.
What happens when this elegant, redundant system catastrophically fails? Let's begin with anorexia nervosa.
Anorexia nervosa is arguably the most complex and lethal condition in psychiatry. From the perspective of the appetite network, we see a profound, terrifying uncoupling of physiological signals from the patient's subjective interoceptive experience.
Explain interoceptive awareness in a healthy person first. How do we normally perceive a meal?
Normally, if we test a healthy individual in a lab using visual analog scales, asking them to rate their subjective feelings of hunger and fullness on a continuous line from 0 to 100 before, during, and after a meal, we see a beautiful, elegant inverse relationship.
The lines cross over perfectly.
Yes. As the meal progresses, the hunger score drops smoothly and linearly, while the fullness score rises smoothly. The subjective mind accurately reflects the physiological reality of the gut.
But in a patient severely ill with anorexia,
the scales disintegrate. If you track a patient with anorexia around a test meal, those subjective percepts fire chaotically and disjointedly. A patient might report that their fullness suddenly spikes to 100 before they have even taken a second bite.
And their hunger score.
Their hunger score might remain chronically high, but completely flatline, never responding to the ingestion of calories.
The dashboard is completely broken.
Precisely. The physiological signals from the vagus nerve and the metabolic systems are still firing. The gut is empty, leptin is critically low, ghrelin is high, but those signals are being violently overridden or misinterpreted by top-down cognitive and emotional interference from the cortex and the amygdala. The network is essentially jamming its own survival signals.
And there's an animal model for this that I found deeply unsettling to read about. The activity-based anorexia model, or ABA. How do you induce anorexia in a rat?
The ABA paradigm exposes a chilling vulnerability in the mammalian brain. To induce it, researchers take a healthy, normal rat and give it unlimited 24-hour access to a running wheel. However, they restrict the rat's access to food to only a single 1 or 2 hour window per day.
Logically, a hungry animal should sleep all day to conserve energy and then eat ravenously during that 1 hour window.
That is what logic dictates. But over just a few days, a devastating paradoxical behavior emerges. The rat will begin running progressively more on the wheel, often logging miles and miles, and during its brief food window, it will eat progressively less.
The hyperactivity suppresses the feeding drive.
The rat enters a feedback loop where it will literally run itself into severe self-starvation and eventually death, despite having food available every single day.
Why would a survival-driven organism choose running over eating when it is actively starving to death?
It appears the severe caloric restriction combined with the physical activity hyperactivates the mesolimbic reward circuitry. The dopamine release associated with the running becomes pathologically highly valued, completely overriding the hypothalamic homeostatic drive to eat.
And the dopamine system is the key vulnerability here, right?
Yes. We know this because of genetic knockdown studies. If researchers genetically modify a mouse to have decreased expression of the dopamine transporter, or DAY, the protein responsible for clearing dopamine out of the synapse, it means dopamine lingers longer and signals more strongly.
And what happens to those mice?
These dead knockdown mice are significantly more vulnerable to the ABA self-starvation protocol. They succumb to the pathological loop much faster. The dysregulation in dopamine signaling traps them in a compulsive behavioral cycle that overrides survival.
That is harrowing. The reward system essentially hijacks the organism. Now let's contrast that severe restriction with bulimia nervosa and binge eating disorder. How does the network fail when the pathology is overconsumption?
The clinical manifestation is entirely different. If you bring patients with active bulimia into a residential laboratory setting to observe their ad libitum eating over weeks, a distinct pattern emerges.
What does it look like?
For a large portion of the day, they will often eat completely normal, appropriately sized meals. Their basic regulatory mechanisms appear intact. But punctuated into that pattern, usually late in the day or evening, they will experience a complete catastrophic failure of the network and engage in massive binges.
We're talking about consuming thousands of calories of highly palatable foods in a single sitting. Is this just a psychological loss of willpower?
Absolutely not. There is profound, measurable biological blunting occurring at the level of the gut-brain axis. We discussed extensively how CCK release and gastric stretch are the primary brakes for a meal. In patients with bulimia, rigorous laboratory studies show that their meal-stimulated release of CCK is significantly blunted compared to healthy controls.
So the gut is literally failing to produce the chemical stop signal.
Yes. Furthermore, their stomachs physically change. Because they routinely accommodate massive binge volumes, the gastric tissue adapts. Their stomach capacity increases, and their sensory sensitivity to gastric distension decreases.
The physical stress receptors, those IGLEs we talked about, are worn out. They require drastically more food volume to achieve the exact same neurological rate of vagal firing that a healthy person gets from a normal-sized sandwich.
Exactly. The brakes are mechanically and chemically degraded. And this peripheral failure is compounded by central neurochemical vulnerabilities, particularly involving serotonin.
Let's dive into serotonin. How do we test its role in binge eating?
We can probe the serotonin system using a fascinating challenge called the acute tryptophan depletion test, or ATD. Tryptophan is the essential amino acid precursor required by the brain to synthesize serotonin.
Okay, so how do you deplete it?
In the ATD protocol, you give a patient a large, foul-tasting protein shake comprised of all the essential amino acids except tryptophan.
What does that do?
It forces the liver to rapidly synthesize proteins, which pulls all the remaining circulating tryptophan out of the bloodstream. This creates a temporary, severe bottleneck in the brain. Over the next few hours, central serotonin levels plummet.
And what happens to the patient with bulimia when you induce the serotonin crash?
If you perform this test on a patient with bulimia who is currently in remission and being successfully treated with an SSRI, the acute depletion triggers intense, overwhelming, and distressing urges to binge.
So it's actively mediating the impulse control.
It strongly indicates that a functional deficit in central serotonin signaling is actively mediating the loss of impulse control and the acute binge urge.
And where specifically is serotonin acting in the network?
It acts widely, but specific receptor subtypes are crucial. Serotonin acting at the 5-HT2C receptor in the hypothalamus is known to promote satiety and inhibit feeding.
And what about impulsivity?
Conversely, alterations in binding at the 5-HT2A receptor, which is heavily expressed in cortical regions, are strongly implicated in the cognitive rigidity, altered reward processing, and impulsivity seen in eating disorders. When serotonin drops, the 5-HT2C satiety signals fade, and the 5-HT2A-mediated impulsivity may dominate.
And we must return to the reproductive axis here, because bulimia is overwhelmingly diagnosed in biological females. Does the hormonal cycle interact with these binges?
Intimately. Binge frequency does not occur randomly across the month. It heavily tracks the menstrual cycle. Prospective clinical tracking shows that binges peak significantly in the mid to late luteal phases.
So when progesterone is high.
This is the precise window when progesterone levels are high, and the protective, satiety-enhancing effects of estrogen we discussed earlier are waning.
The hormonal armor is removed, just as the emotional distress of PMS might be rising.
Exactly. Furthermore, translational animal models suggest a critical developmental risk factor regarding estrogen. If researchers perform a oophorectomy on female rats before puberty, surgically removing their ovaries and entirely depriving them of the normal surges of pubertal estrogen, those rats show a significantly higher lifelong risk of developing binge-eating traits when exposed to palatable food.
So prepubertal estrogen doesn't just regulate immediate feeding. It appears to permanently organize the brain circuitry to be structurally resilient against binge eating in adulthood.
That is the leading hypothesis. It highlights how vulnerability to an eating disorder is woven into the neurodevelopmental timeline long before the first diet is ever attempted.
So what does this all mean? As we arrive at our integration and clinical reasoning section, how should the psychiatrist, the trainee, or the medical student listening to this integrate all these densely packed moving parts into their daily practice?
The essential synthesis for the clinician is a radical shift in perspective. You must fundamentally and permanently abandon the paradigm of willpower. Eating disorders and severe clinical obesity are not moral failings, nor are they simply psychological neuroses.
They are measurable physical dysregulations.
They're complex, quantifiable dysregulations in a highly redundant, structurally integrated neurohormonal network.
When a patient sits in your office and describes the shame of a massive bulimic binge, you have to see past the behavior.
You must. You cannot just see a behavioral choice. In your clinical mind's eye, you must see the blunted release of CCK failing to signal the NTS. You must visualize the altered 5-HT2A and 5-HT2C receptor binding failing to regulate the patient's impulsivity.
And you must recognize the luteal phase hormonal shifts that are actively amplifying their neurobiological vulnerability on that specific day.
Our job as psychiatrists is to understand the intricate mechanics of this embassy, to use our clinical history to figure out which diplomats are shouting and which have been structurally silenced, and to target our psychopharmacological and behavioral therapies accordingly.
That is an incredibly empowering, destigmatizing paradigm shift. Before we move to the learning reinforcement, I want to leave you, the listener, with a final provocative thought. Something to mull over as you walk away from this deep dive. We have established today that our food preferences are overwhelmingly learned.
They are.
We've also established that our delicate, ancient satiation signals, like the gentle mechanical stretch of the stomach and a tiny chemical pulse of CCK, were designed to stop us from eating fibrous fruits, lean game, and seasonal berries.
But these ancient signals can be so easily overridden by the sheer engineered variety and intense hedonic reward of ultra-processed, hyper-palatable modern foods.
The dessert stomach mechanism was meant to ensure we ate both berries and nuts. It was never meant to navigate a buffet of engineered pastries. So the question is, are our ancient brainstem and hypothalamic circuits fundamentally, structurally incompatible with our modern industrialized food environment? We are running Paleolithic software in a 21st century hardware store.
That's a great way to think about it.
And if that is the case, how much of psychiatric intervention in the future will have to evolve? Will we move beyond just treating the psychological symptoms of distress and begin utilizing targeted interventions to directly retrain and restructure the gut-brain axis itself?
I believe it is the defining existential question for the future of nutritional psychiatry and metabolic medicine.
To lock in the clinical reasoning from this intensive deep dive, let's test your mental models with 5 quick, single best answer questions. I'll read the clinical or physiological scenario, give you a moment to think about your answer, and then we will break down the precise mechanism. Here is question 1. A dietitian advises a patient to consume a large bowl of vegetable soup rather than a dry sandwich and a glass of water, noting it will keep them full longer. Which specific neurophysiological mechanism best explains why incorporating liquid into the food matrix increases the satiating effect? I'll give you a second to think. All right, what's the answer?
The answer is the synergistic firing of vagal afferents in the nucleus tractus solitarius. The soup creates prolonged gastric distension, activating the IGLE mechanoreceptors. When that sustained physical stretch is simultaneously combined with the chemical release of CCK triggered by the nutrients in the soup, the vagal action potentials traveling to the brainstem are not just added together, they are exponentially, synergistically amplified.
Brilliant. Question 2. A young patient with a BMI of 14 due to chronic severe restrictive eating anorexia nervosa is placed on a careful inpatient refeeding protocol. From the perspective of evolutionary neuroendocrinology, how is the hormone leptin functioning in this patient's hypothalamic circuitry? Take a moment to think about it. Okay, let's see the breakdown.
In this severely emaciated patient, the profoundly depleted circulating leptin levels are actively removing the normal inhibition on the TEAM-GO neurons and simultaneously failing to stimulate the TEAM-STOP neurons. This massive shift in the arcuate nucleus creates an intense, biologically driven negative reinforcement hunger, specifically designed by evolution as a desperate alarm to defend the organism against imminent death by starvation.
Spot on. Question 3. In patients actively suffering from bulimia nervosa, rigorous laboratory studies demonstrate that typical physiological stop signals are severely impaired during a meal. Which specific hormonal blunting is classically observed during the ingestion of food in these patients compared to healthy controls? Think about your answer. All right, what's the mechanism here?
The classic finding is a significantly blunted meal-stimulated release of cholecystokinin, or CCK, from the small intestine. This lack of the primary chemical break directly leads to delayed and weakened postprandial subjective satiety, contributing to the massive volume of the binges.
Question 4. Following a massive holiday feast, a healthy individual states they are stuffed and in physical pain, yet they readily accept and consume a large slice of rich chocolate cake. Which two specific neural structures are primarily responsible for encoding the reward and generating the intense positive hedonic response that violently overrides the vagal stress signals from the gut? Take a second to recall the structures. Okay, what are they?
The dopaminergic pathways in the nucleus accumbens encode the wanting and incentive reward value of the sweet stimulus, while the opioid and endocannabinoid signaling in the ventral pallidum actively generates the subjective conscious liking or pleasure. Together, they act as the central subcortical nodes for sweet taste reward that override homeostatic satiety.
Excellent. And the final question, question 5. A new, highly targeted anti-obesity medication is prescribed to a patient. The mechanism of action states that the drug acts as a direct agonist at the melanocortin-4 receptor, or MC4R, in the downstream targets of the hypothalamus. Which endogenous molecular signal is this synthetic drug chemically mimicking to suppress the patient's appetite? Take a moment to formulate your answer. All right, what's the verdict?
The drug is mimicking alpha-melanocyte-stimulating hormone, or alpha-MSH. Endogenous alpha-MSH, released by POMC neurons, normally acts as an agonist at the MC4R to inhibit eating and increase energy expenditure, acting in direct competitive opposition to the functional antagonist AGRP.
Excellent work. We strongly encourage you to head back to the textbook, review the dense neurobiology, read alongside the chapter, and continue deepening your clinical understanding of these incredible pathways.
Here are the details of this episode.
PSYCH 017. Basic science of appetite.
Filing.
Hunger and desire, how the brain regulates need, reward, and restraint.
Episode description. Appetite is often mistaken for a simple biological drive, but it is in fact a finely regulated system integrating energy balance, reward, emotion, and cognition. This chapter explores how the brain determines when to eat, what to eat, and when to stop.
In this episode, we examine the interplay between homeostatic systems, which monitor energy needs, and hedonic systems, which assign pleasure and reward to food. The hypothalamus plays a central role in maintaining balance, responding to hormonal signals such as leptin and ghrelin. At the same time, reward circuits involving dopamine-shaped motivation and craving.
We explore how appetite is influenced by context, emotion, and environment. Eating is not simply about energy. It is embedded in social, psychological, and cultural frameworks.
Dysregulation in these systems can lead to a range of psychiatric and behavioral conditions, from eating disorders to obesity and addiction-like patterns of consumption. These are not failures of willpower, but alterations in the systems that regulate need and reward.
This chapter reframes appetite as a negotiation between biological necessity and experiential desire, revealing how the brain balances survival with meaning. Key takeaways.
Appetite is regulated by both homeostatic and hedonic systems.
The hypothalamus monitors energy balance and responds to hormonal signals.
Hormones, such as leptin and ghrelin, influence hunger and satiety.
Reward systems, particularly dopamine pathways, shape food-related motivation.
Appetite is influenced by emotional, social, and environmental factors.
Dysregulation can contribute to eating disorders, obesity, and addictive behaviors.
Eating reflects both biological need and psychological meaning.